Hello, my name is Hugo Layard Horsfall. I am a neurosurgery trainee in London. I started off in the tech group in 2020 as an SHO junior clinical fellow year. Subsequently got a number in London, and I've been doing research with Hani and the rest of the tech group for the last 5 years. So at the moment I am currently out of neurosurgery training and I am doing a PhD at UCL and the Francis Crick Institute, supervised by Andreas Schaefer, William Merhead and Hani Marcus. My project looks at novel brain activity at the single unit level. So we're using state-of-the-art electrodes known as neuropixels. These are small devices which are about half the width of a human hair, and they are implanted temporarily into the human cortex, which is about 1 centimetre in depth. Lots of this work started in 2017, where they developed these probes for animal research and were able to find fundamental neuroscientific paradigms about how the brain functions in various disease and healthy states, and how neural activity is indicated in behaviour, integration of movement and also cognition. One of the most exciting aspects of this research is the unknown. This is very exploratory research. Traditional mechanisms or techniques to understand neural activity have been non-invasive. So this is functional imaging such as functional MRI, or you have non-invasive electrophysiological imaging, monitoring such as scalp electroencephalograms, which is where you have scalp electrodes placed at various parts of the head to understand groups of neurons and how they fire. So what's really exciting about neuropixels is, although it's invasive, we take appropriate steps and measures to protect patient safety. By inserting this electrode into a human cortex during one of their operations, we can record from an individual neuron and understand how that neuron communicates. And ultimately, the long-term goal of this is exploratory neuroscience to understand how neurons communicate in various disease states such as Alzheimer's, or for example, if someone has a tumour, there are different types of tumours, some are very aggressive, some are less aggressive. What is the electrical difference in these tumours? How does electrical activity relate to the underlying genetic and molecular characteristics? But also from a cognitive point of view, how do humans make decisions, and more broader kind of neuroscientific findings. So as always, this is massively multidisciplinary. It's supported by UCL, the Francis Crick Institute, and it's only feasible in certain institutes where all of these different factors come together, really with the patient at the heart of the research. A lot of the challenges are novel devices, regulatory considerations, proving that the device is safe and feasible. That's a challenge, I think, for lots of neurosurgical innovation. So it's finding ways to understand these and mitigate them within the confines of still pursuing research. This neuropixels work falls under the broader spectrum of neurotechnology, which is a specific research stream within the tech group. And neurotechnology is an interesting term. It's growing in scope, both in terms of media coverage and devices available to clinicians and patients. People might have also heard about brain-computer interfaces, which are essentially devices that can decode and record neural activity from the brain in a variety of different ways, and that information then goes to an output device. You have different ways of accessing the brain. Some are non-invasive, some are implantable, and some are accessible through the vascular system. So notable companies, which I'm sure many people have heard of, are Synchron with their Stentrode and Neuralink with the N1. And more recently, there's Precision Neuroscience based out of New York, which has a device which sits on the surface of the brain. And these devices are interesting because they record neural activity. They have slightly less resolution, for example, than the NeuroPixel probe, but NeuroPixel probe can only be inserted temporarily and is very fragile. So with everything in kind of medicine and innovation in neurosurgery, there's a trade-off between these capabilities of these devices the long-term implanting ability, and ultimately what the output is. So whilst brain-computer interfaces at the moment are much more interested in helping people with paralysis or locked-in syndrome to use a robot effector or be able to control an iPad just using their thoughts alone, which is massively important and is a really exciting area of research. The NeuroPixels probe and the research that we're doing is very exploratory and kind of discovery science to understand how we can understand the central nervous system in a much more profound way and integrate that with different molecular characteristics or imaging modalities to get a deeper understanding of the brain.